I'm happy to be here today to talk about the viral vector CDMO business at Oxford Biomedica. For those of you who are not familiar with the organization, Oxford Biomedica was founded in 1996 and was listed on the AIM in 1996, and it was listed on the London Stock Exchange in 2001. We have headquarters in Oxford and locations outside of Oxford in both Dublin and in the US, but the bulk of our manufacturing is based in the UK. Just a little bit about us. We have integrated offering, so from the gene of interest all the way through process development and on up through manufacture of 50, 200, or 1,000 liter scale GMP grade viral vectors. We are a leader in viral vector manufacturing of both lentiviral vectors as well as adenoviral vector-based medicines and other viral vectors as well. We have a very collaborative model. We either can bring a project that's been tech transferred in and we make the partner's product as prescribed by them, or we can take their gene of interest and pop it into our platform, our lentivector platform, or we have other abilities to work together to develop new platforms. And we have been approved by many regulatory agencies around the world. We make product that is distributed into 28 countries so far, commercial product that is. And we are also an engine of innovation. We are innovating both on our lentivector platform as well as on general viral vector production, both upstream, downstream, analytical testing, and all aspects of viral vector manufacturing. We have several long-term partnerships that we're very proud of. The companies listed on the top there, those are our partners for our lentivector platform. So you'll notice there are several pharma companies there. We are now partnered with Bristol Myers Squibb. We've been partnered with Novartis for quite a while. We proudly make the viral vector that is used in Kymriah for Novartis. We've been working with Novartis since shortly after they obtained that technology or attained access to that technology through the University of Pennsylvania, and we very proudly make that vector. And several other companies there you see. Sayo, we are also very proud. Their lenti product to address Parkinson's disease was actually developed by us, out-licensed to them, and we continue to make the vector for that product. And then we also have several tech transfer programs that we're proud to share. The AstraZeneca, which I'll be talking about a little bit later with their COVID vaccine, as well as programs with Boehringer Ingelheim. So some key considerations when thinking about developing cell and gene therapies. Streamlined product development is really the key to getting to clinical material quickly. And the ability to leverage an existing platform really helps to streamline that process. So the ability to pop a gene of interest into established platform for which there are already analytical methods established, protocols written, materials validated, this really can make the process a lot simpler for a new company that's coming into the space and wants to get to the market quickly. So identifying partners with established experience is really going to be the key to speed. And also safety considerations, so identifying an appropriate dose, determining testing, being able to leverage some of the experience that have already occurred with products coming off of that platform can help cut corners, if you will, be able to leverage some of the experiences that are, not have to reinvent the wheel, basically. When thinking about commercializing cell and gene therapies, economies of scale is extremely important. And I'll just tell you right now, the spoiler alert there is suspension culture. Because if you can manufacture suspension culture at small scale, you can manufacture suspension culture at virtually any scale. And so that will allow you to obtain economic viability at the scale that is needed for the product. So accelerated regulatory pathways, which a lot of products in our industry are going for, so these breakthrough, fast track, RMAT designations, they allow for compressed timelines with engagements with regulators, which is great. But in order to actually meet those timelines, you gotta bring it too, right? You gotta be able to bring the data to them in the timeline that's been compressed. And the best way to be able to do that is to be able to leverage platform and data and experience from existing products off of that platform. And the overall goal is to get the patients to the product quickly. That's what all of this is about. That's what we all want. That's why we're all here. So we combine all of our experience in lentiviral and gamma retroviral therapy based medicines, as well as now adenoviral vector-based medicines. And so I think this is a bit of a surprise, I think, for a lot of people, because I think Oxford Biomedica has been synonymous with the Multivector Company for a really long time. And we are, and lenti is an important part of who we are. But we are now a multi-vector company, not just solely focused on lentiviral vectors. So we combine all of our experience with the viral vectors, the upstream, the downstream, the analytics, and all of those important technical considerations, combined with all the functional groups required within a CDMO to really bring success for our partners. And to be honest, when we think about the functional groups within a contract manufacturer, it's worth mentioning our client program and alliance management, or our CPAM group, because our partners always do. This is our group that serves as the internal advocate and champion for our partners within the organization, and they are top-notch, really making sure that the program stays on time and on budget, and that everybody is getting what they need and everybody is happy. They are the peacemakers. They are the front line. Also worth mentioning is our quality and regulatory leadership team. So we have, I mentioned earlier, we have this location in Ireland. This enables us to be able to release product through into Europe, as we are in the UK. And also our quality group and regulatory group come together to make sure that we have been able to generate product that is going into 28 countries. So that's many regulatory agencies for which we've had to work with. And I mentioned this earlier, but I'd like to take a minute just to talk about, the different collaboration models we have. So all the way on the right, our LentiVector platform, where partners can come to us with their gene of interest. It can go into the platform, and then it's a simple, the manufacturing is simplified because of the experience we already have with that platform. And it's just an out license of our platform, as well as manufacturing of the product, leveraging the experience and expertise that we have. All the way to the other side is a technology transfer project where the client comes in with the protocols, the product, the materials, and everything, and we just make it to their specifications. And then in the middle is the ability to really be collaborative and put our heads together and bring the novel emerging technologies that are coming up in the space and combining that with the expertise and experience that we have in order to make something new and exciting. And we're more than happy to do that and delighted to have several programs along that line. And so I know that this is a cell and gene meeting on the Mesa, but I want to, if you'll humor me for a moment, I'd like to take a bit of a left turn for a second and talk about COVID vaccine. Some of you may or may not have heard, while we were all relegated to our home offices over the last nearly 2 years, that Oxford Biomedica has actually been making COVID vaccine for AstraZeneca, and this vaccine is approved and being distributed and used in the UK and other countries around the world, although it's not approved here in the US yet. So I'd like to take a minute to explain how this came about, because it really does harken back to cell and gene, I promise. So we all know the COVID timeline, right? So back in the spring of last year, Oxford University approached us with this vaccine candidate that they had for COVID. And the reason why they were able to have it in the spring of last year already is because they were using this ChAdOx1 vaccine platform that they had discovered and described in 2012. And they had actually developed this platform for MERS, Middle Eastern Respiratory Syndrome, which is a sister of COVID. And so when COVID happened, they actually retooled that vaccine quickly to address COVID. They came to us, asked us to make the vaccine for them. And then things moved very quickly. AstraZeneca licensed it from the university, and at the same time, AstraZeneca came to us directly asking us to make the vaccine now for AstraZeneca. So that was in May of 2020. So also should mention an important part of that is the fact that we had a brand new facility come online in January of 2020. And as any good manufacturer knows, when you bring a new facility online, you have a lot of fallow space. So here we had this new, gorgeous, high-tech facility for cranking out GMP-grade viruses, effectively. And now here's this big need for a viral-based vaccine. And so we started ramping up, our team to be able to make the vaccine. And by the end of last year, we were cranking out 1,000-liter batches of the vaccine. And it's important to note that we had never made 1,000-liter batches of anything before last year. And we had also never made adenoviral vector-based medicines before. However, our experience with suspension-based lentiviral vectors made it easy for us to adopt the knowledge, experience, equipment, facilities that we already had to be able to do this successfully. So I think to bring it back to cell and gene therapy, if we look for a moment at sort of the history of gene therapies and think about, when gene therapies were first developed and being brought online, you had the, we were founded in 1996, right there, kind of in the middle of all of that. And then, as we all know, Jesse Gelsinger in 1999, there was a bit of a slowdown for about a decade, and then things started picking back up. Glybera is approved in 2012, and then in the last 5 years, all these products have been approved. If you look at all of these products that have been approved and all of the indications that they are addressing, all of them are addressing rare diseases. None of them are addressing an indication for which 1,000 liters will ever be needed. And so I think that in the field right now, there's this big question mark of, will we be able to manufacture viral vectors at scale when the time comes? And our experience with the COVID vaccine tells us, at least, that we know we can. So when that gene therapy comes that's going to address a blockbuster indication, we know that we're ready. So why partner with Oxford Biomedica? We're customer focused. We are leaders in lentiviral vector as well as adenoviral vector commercial manufacturing and other viral vectors as well. And we are ready. Thank you for your attention. So I think we have time for a few questions if there are any. Oh, there's a question. Is there a microphone? Oh, he's coming.
Okay. So I think from what I hear, the variability in the produced vectors is really one of the major challenges in cell and gene therapy. What do you think would need to happen in order to solve this issue?
The viability of the vectors?
The variability. The variability.
Oh, variability of the vectors. Which one in particular, or just in general? I think there are unique challenges associated with the different viral vectors. It'd be hard to answer that, but I'd be happy to talk to you afterwards, because there are unique challenges with the different ones, and we could talk about that. Okay, you want to cut it off? Or one more minute for another question? Maybe one more question? No? I don't think so. That's okay. I'll be in the back if anybody has questions they want to come talk to me.